Okay, so our second speaker this morning will be Dr. Cheryle Seguin. Cheryl came to the University of Western Ontario about 2 years ago, I guess, after being recruited from Sick Kids, where she did a postdoc with Dr. Janet Rossant. She's going to, her research is focused on approaches to manipulate stem cell fate to generate lineage-specific progenitor lines from human embryonic stem cells and human-induced pluripotent stem cells. And she's going to talk to us today about her talk defining pathways to regulate stem cell fate. So please join me in thanking Cheryl.
Well, thank you very much. As I'm giving the talk, I'd like to exciting it is to be included in such a prestigious lineup, and also how lovely it is to follow Derek. A lot of the work that Derek's lab has done has actually really informed on the strategies that my lab takes in trying to understand how to control stem cell differentiation. So as Sean mentioned, I've been here at Western since 2009, where I've started a few studies looking at how we can engineer differentiation. And so I'll present to you today two of the stories that we're currently working on. And so my lab looks at the embryo and thinks a little bit about the pathways that control how these pluripotent cells become specified during development. And so I'm sure we're all aware of the tremendous potential of embryonic stem cells. Since their inception, they've been widely cited for their potential to differentiate into any cell type in the human body. And what we've been focused on in work that started during my postdoc with Janet Rossant was how cells actually undergo this transition and what pathways are initiated, what cascades get turned on, and how perhaps we could learn from development to control stem cell differentiation for potential application. Because while the field is tremendously excited by the potential of these cells to become every cell type in the human body, it's also a great challenge, because if what as a scientist, if we're trying to make perhaps pancreatic cells, we don't want to accidentally, as Derek might have just outlined, make brain, or make something else that, following transplantation would be quite detrimental to the patient. So we think it's quite important to understand these pathways and then perhaps use that knowledge to guide stem cell fate. So as I mentioned, there's two stories that I'm going to tell you today. One is a series of experiments that looks at quite early development and how extra-embryonic cell types actually contribute to patterning the embryo. And then the second story will be some recent work that we've done in my lab to try to generate pancreatic progenitors from embryonic stem cells. So we'll start in the very early embryo. And so work by my previous mentor, Janet Rossant, was instrumental in understanding all of the stem cells in the early embryo. So not just the embryonic stem cells that come from the early epiblast, but also the extra-embryonic cell types. So these cell types don't actually give rise to the embryo following development, but they play quite instrumental roles in both supporting and patterning early embryonic development. And so one of the very interesting cell types to us was the extra-embryonic endoderm. So this cell type is an early progenitor cell that later stages in development actually goes on to give rise to both the visceral endoderm and the parietal endoderm. And while this external layer is important in establishing the yolk sac and establishing maternal-fetal nutrition, the visceral endoderm lies right adjacent to the developing embryo and actually plays very important functions in terms of signaling. So the very first thing that it does is it's quite instrumental in specifying mesoderm. And so signals that derive from this visceral endoderm are quite essential for mesoderm patterning and then the subsequent development of the downstream pathways. Another important structure that derives from the extra-embryonic endoderm is the anterior visceral endoderm. And this plays very important roles in neural induction and the maintenance of head character. And so we wanted to understand how this signaling happened. And so it's essential in the early embryo. We know that if you lose extra-embryonic endoderm, the embryos can't develop. And we wanted to try to come up with an intriguing strategy to model this in culture. So previous studies had used similar cell types that were endoderm-like to try to understand this interaction between the embryo and the extra-embryonic tissues. But in working in Janet's lab, we had access to stable cell lines that were derived from the mouse embryo. And so some of the early work that I did in Janet's lab was deriving Zen cells, or extra-embryonic endoderm cells, directly from the mouse embryo at about embryonic day 3.5 or 4, and developing systems to understand how these two cell types actually communicate in vivo and seeing if we could recapitulate that in the dish in vitro. And so as I mentioned, the first system that we started with was a direct cell culture model. And so what we did is we derived extra-embryonic cell types from green fluorescent mice so that we could trace their development, and paired them with reciprocally labeled embryonic stem cells to see if we could both follow these cells fluorescently in the dish and see if we could recapitulate this sort of cell-cell interaction that was happening in the early embryo. And so in direct cell culture, we essentially used the extra-embryonic cells as a feeder layer. So we laid a monolayer down of extra-embryonic endoderm cells, and then on top of that seeded the embryonic stem cells to see if we could mimic development. And initially, hopefully you can appreciate, they form small ES-like colonies that are surrounded by these extra-embryonic cell types. So here they're marked fluorescently in red, and you can appreciate these rings that surround the cells. And we used an embryonic stem cell line that was marked where GFP was expressed from the FLIC1 promoter. And this enabled us to watch mesoderm differentiation happening in culture. And hopefully, it projects better than I can see, we can see that following co-culture, following 8 to 10 days in culture, we start seeing a pretty robust mesoderm induction in these cells. And so we were hoping that this was modeling what was happening during early development. So we looked in a couple of different ways. We used some antibody staining, comparing ES cells grown with the extra-embryonic endoderm to ES cells just grown on normal feeder cell layers, and we're able to observe the induction of mesoderm differentiation through NKX 2.5. We had PCAM positive structures formed in the dish, and cardiac troponin T positive cell clusters, which is a marker of differentiated cardiac cells. Now this will be the big. And of course, the big marker of cardiac differentiation is beating cardiomyocytes. And thankfully, the movie is playing. And it's always really great when you get a phone call on a Sunday from your student because the cells in dish are beating. And so we were quite happy to see. And here you can see this is the last few red Zen cells. So this shows you that the beating structures are actually ES-derived, not extra-embryonic endoderm-derived. And so in spontaneous ES cell cultures, you don't see beating cells when you grow them in monolayer until you take them out to about day 21 or so. In contact with the Zen cells, after 12 days in culture, most of the dish starts beating. And so we were quite pleased that we could recapitulate the cell interaction, and now we wanted to understand what was mediating it and what some of the pathways behind it were. And so we developed a second strategy to try to do this, and this time instead of direct cell culture, we did an indirect way to figure out if this was cell contact dependent, where essentially we just took the media conditioned by extra-embryonic cells, put it on top of our ES cells and let them differentiate in culture, tried to assay if we could recapitulate induction of cardiac differentiation one more time. So again, here, what we're comparing is ES cells grown just in media without LIF. And so for mouse cells, you need to put leukemia inhibitory factor on your cells to keep them undifferentiated. When you withdraw it, they undergo spontaneous differentiation. So what we were hoping to compare was the spontaneous differentiation to that induced by the extra-embryonic cell types. And hopefully you can see here, again, using the ES cells marked with the FLIC1 or mesoderm reporter, that we can see induction of mesoderm differentiation in the presence of extra-embryonic factors. And this doesn't happen in quite the same temporal way when the cells are just undergoing spontaneous differentiation. So a very talented undergraduate student, actually, in my lab, undertook a series of very specific gene expression analyses to try to see if he could identify the window of induction and compare these two things. So here you're looking at some real-time PCR, comparing in red, Zen conditioned media, to just spontaneous differentiation. And what he was able to show quite nicely is that the extra-embryonic endoderm-derived signals promote mesoderm induction more than spontaneous differentiation, also induction of markers of cardiac differentiation, and that this was somewhat specific. So we also looked for induction of hematopoietic markers or endothelial markers, which were not induced more than spontaneous differentiation. So it seemed that these factors that were being secreted by the extra-embryonic endoderm were specific for cardiac induction and not any mesoderm-derived structure. So again, oh, not as lucky this time. You'll have to maybe just squint a little bit and pretend this is actually beating too. And so we were able to recapitulate that cardiac induction that went all the way through to beating cardiomyocytes in the dish just by growing cells in the presence of this conditioned media. So subsequent to these studies, Ryan was quite keen on understanding perhaps what the signals were and how cells were responding to these. So he set up a set of experiments using a variety of different signaling and pathway inhibitors, trying to really understand how these signaling cells, in our case the XEN or extra-embryonic endoderm cells, were in fact secreting signals into the environment and what pathways might have been induced in the ES cells to receive these signals. And so a wide range of experiments was set up, and Ryan spent quite a bit of time screening inhibitors and trying to change this induction. So I'll just show you some of these studies. Essentially what you're looking for here in this real-time PCR data, again looking quite at specific time points over this 12-day period. So the conditioned media experiments are in red. So what he was looking for is anything that would promote over the red line or anything that would prevent differentiation using these molecular inhibitors. So your reference is always this red line. And by looking, what Ryan was able to show is that the ERK and P38 pathways are required in ES cells to undergo mesoderm induction. And that in fact if you inhibit the JNK pathway or use a JSK inhibitor, which promotes Wnt signaling, that you can actually promote mesoderm induction in ES cells. So this was the early window of differentiation, and Ryan was quite excited to look at what would happen at later stages. And so he did the same set of experiments, but this time looking at the later stage markers of cardiac differentiation. And again, what we're looking for is anything that changes, deviates from this red line, which is just XEN conditioned media. And what Ryan was able to show is that the P38 pathway is absolutely required for cardiac differentiation. So hopefully you can appreciate that brown line, which is P38 inhibition, is really just flatlined. And so if you put a P38 inhibitor on there, you prevent all cardiac differentiation. And what he also showed is that inhibition of JSK signaling, or Wnt signaling, actually promotes cardiac differentiation. And here what we've done is outlined areas of contractile beating in these cells. And whereas normally it takes about 12 days for cardiac induction to happen, if we use a GSK inhibitor, we can promote beating by day 8. So we're able to move that sequence forward and actually change cell differentiation. And the last bit of the story here, which was always really quite interesting to me, is this communication reciprocal? So we know that the extra-embryonic endoderm actually signals to the undifferentiated portion of the embryo, but we are quite intrigued to understand what signals derived from the embryo would do to the extra-embryonic endoderm. Because if you remember back to the schematic, the extra-embryonic endoderm can become either parietal or visceral endoderm. And we had the notion that juxtaposition with the embryo would actually promote visceral endoderm differentiation. And so we set up a set of experiments. I won't go through all of them with you, but essentially what we were able to show by both direct cell contact and through conditioned media is that the signals that come from the embryo are actually quite instrumental in patterning the extra-embryonic to become visceral endoderm. Okay, so that was the first set of experiments where we were looking quite early in development. Now, the second set of experiments that I'll quickly go over have to do with understanding signal transduction and the activation of transcriptional networks. And so what we essentially did in Janet's lab, when I walked in the door, I was told to go make human extra-embryonic endoderm, because this had never been done before, and this is what Janet's lab studied. And so she essentially sent me to the literature to understand early human development, to say, come up with some transcription factors and let's try to engineer development. So we had a whole bunch of tools in the lab that enabled us to drive gene expression and create transgenic cell types. And so I was charged with this notion of trying to take the undifferentiated cells and make them extra-embryonic endoderm. So we were way up here in the embryo when I initially started these studies. And so this is a set of experiments that we published in Cell Stem Cell in 2008, and on which my lab has kept going. And so we started with this notion of candidate transcription factors and settled on the SOX family. And so SOX7 and SOX17 are quite intriguing in their expression patterns in the early embryo. SOX17 is expressed both in the extra-embryonic endoderm, but also coincident with the differentiation of the definitive endoderm. SOX7, on the other hand, its expression is restricted in the mouse, at least, up to the extra-embryonic endoderm. When we look at knockout animals, we know that if you remove SOX17 expression, there's no phenotype in the extra-embryonic tissues, but that there's a total lack of definitive endoderm differentiation. And so we had the hypothesis when we started these studies, working with Andres Nagy at Mount Sinai, was that if we overexpressed either SOX7 or SOX17, we would make extra-embryonic endoderm progenitor cells. And so this was the first set of experiments that I conducted in the lab. So we generated human ES cells using a Cree-inducible system where we could introduce these plasmids into human ES cells but only turn them on using Cree. To figure out if the SOXs could guide cell differentiation. So here is an undifferentiated cell colony, and hopefully you can appreciate that SOX7 expression induced a major change in these cells. They had a remarkable change in cell phenotype. They started growing as flat monolayer type cells, and we had very robust expression of SOX7 in these cells. In parallel, we generated cells that overexpress SOX17, and on initial examination, we were quite let down because there was really no change in cell morphology. So these cells that overexpress SOX17 still grew as colonies. The cells perhaps were a little bit less tightly associated, but there was no overt change in cell morphology. Luckily, we kept going with these. So these are the SOX7 cells, and we did some antibody staining to try to determine what markers were being induced and what differentiation was going on. So here are the undifferentiated ES cells on this side, and those are actually supposed to look like black boxes. However, on this side, when we, thank you, Sean, overexpress SOX7, we can see the SOX7 is nuclear. We can see that SOX7 expression turns on SOX17, which is quite intriguing to us, and also GATA4 and alpha-fetoprotein, which are markers of the extra-embryonic endoderm. Sorry about that one, not sure what's happening there, but one of the really intriguing things about these cells is while they acquired markers of differentiation, they never lost markers of ES cells. So we still had high expression of OCT and NANOG in these cells, which had some of the reviewers being quite skeptical about our data. So one of the things we did was show that these markers of differentiation, in this case AFP, was actually co-expressed with OCT4. And so what we feel is that these cells are maintaining some level of pluripotency markers that are making them progenitor cells, so still able to renew. When we look at the SOX17 cells, we see nice nuclear expression of SOX17, the induction of GATA4, one of those pan-endoderm markers. And again, in these cells, we maintain expression of OCT4, but it is co-localized to GATA4, the marker of endoderm. So we did a variety of assays to try to understand what these cells actually were, one of which was microarray analysis. So we identified a large set of probes that was upregulated in the SOX17 overexpressing cells, and this included known markers for definitive endoderm differentiation, a large set of genes that was upregulated in the SOX7 cells, which included known markers of the extra-embryonic endoderm, at least in mouse, and then this set of probes that was common to both, which thankfully included the usual suspects of shared endoderm markers. So we were quite happy that at least in the dish, these cells were behaving like endoderm cell types. This again is just some real-time gene expression showing that the SOX7 cells were expressing markers of the extra-embryonic endoderm, but not the SOX17 cells, and reciprocally the SOX7 cells don't express markers of definitive endoderm, but the SOX17 cells do. So the next question is how they would behave in vivo. So we did some standard teratoma assays. So if you inject a NOD-SCID mouse with human embryonic stem cells, their pluripotentiality is demonstrated by differentiation to cell types of all three lineages. But our question was, what would happen when we injected our genetically engineered cells? And in the SOX7 background, we saw differentiation into just large cystic spaces, which would be reminiscent. We were thinking of extra-embryonic endoderm type membranes. And the SOX17 cells generated only mesoderm and endoderm structures with no neuroectoderm differentiation apparent whatsoever. And so we were quite excited at this time that we thought we had created a cell population where we had limited potential. So SOX7 promoted differentiation to an extra-embryonic-like cell type, whereas SOX17 promoted differentiation to what we thought was definitive endoderm. We wanted a functional assay. Now, when you're working in human embryonic stem cells, you learn quite quickly that functional assays are rare in human cells, especially when you're talking about progenitor cells. And so we had to be a little bit creative in how we undertook this, especially so with the extra-embryonic endoderm. It's quite difficult to find a functional assay for an extra-embryonic cell type, but our work in the mouse had actually just provided one. So we went back to this co-culture model, where we had just shown quite convincingly, we thought that the extra-embryonic endoderm could pattern ES cells. So we asked if the SOX7 cells could act just like the mouse cells and pattern ES cells, and that's quite beautifully what happened. So here again, we're just comparing, this time, red-labeled mouse ES cells, and we're co-culturing either with embryonic stem cells and direct cell culture, where we see no induction of mesoderm differentiation, and in fact the cells stay beautifully pluripotent. But if we grow them in the presence of the SOX7 overexpressing cells, we see induction of FLIC1 expression, cardiac troponin T, PCAM positive structures, a loss of pluripotency, and we get the induction of contractile cardiomyocyte differentiation in these cultures. So along this middle axis here. So we were quite pleased that we had shown cardiac differentiation in these cultures, and that SOX7 did promote functional extraembryonic cell types in the human. Now, when it came to proving that we'd made definitive endoderm, we looked at the literature, and at the time a variety of growth factor-induced differentiation protocols were being developed to try to promote hepatocyte differentiation or beta cell differentiation from pluripotent ES cells using stepwise protocols and a combination of different growth factors to get from undifferentiated cells to a functional endoderm cell type. And the intriguing thing was that all of these protocols required an initial specification step to make definitive endoderm. So whether it was EB or active and mediated, if you didn't first make definitive endoderm, you couldn't get to any of these functional cell types. So we figured, okay, we would put our cells through these same sort of assays, skipping the first step, and if we truly had made definitive endoderm, we'd get to the end of both of these. And so what we showed was this was true for hepatocytes, that if you skip the first step with human ES cells, you get no albumin positive cells at the other end. But if you skip the first step and use SOX17 cells, you get quite a beautiful population of albumin positive cells, suggesting hepatic differentiation. And the same was true for growth factor-induced protocols to make beta cells. And so if you omit definitive endoderm differentiation from ES cells, you get very little insulin production. However, the SOX17 cells actually robustly produce insulin-positive cell clusters in these assays. So this is when I left Janet's lab then with this ability to make definitive endoderm, and really with the notion that if you use transcription factors to engineer differentiation, you could do it quite specifically. So we had stable cells, they were still proliferative, so they were progenitor cells, and they were undergoing a pretty homogeneous cell differentiation because we had changed their profiles of gene expression. And so in starting my own lab, what we were looking for was trying to take this one step further to try to promote differentiation of pancreatic progenitor cells, similar to the ones that Derek had discovered in the mouse and in the human. So what we looked to then was development and mouse development to try to come up with a list of candidate genes that seemed logical in their ability to promote or start this cascade of differentiation that might get us to a pancreatic cell type. And so we devised strategies then in the lab to do exactly this. We had a list of candidate transcription factors, and our notion was that if we overexpress them after SOX17 differentiation, that we might eventually get to what we were going to call a pancreatic endocrine progenitor. And so this is a set of experiments done by a graduate student in my lab. And what we wanted to start with first then was defining the window of differentiation. And so we knew that SOX17 wasn't always expressed in the embryo, that it was shut off after definitive endoderm specification, after which other transcriptional cascades were being induced. And so we developed a system then to transiently express SOX17 using the ERT2 system. And hopefully you can see here, so this is a lovely system where the transgene is always expressed, but its activation is dependent on the addition of tamoxifen and its cellular localization. And so in the normal instances, the cells constitutively express this transgene, but it's not nuclear. It's held out in the cytoplasm, and within hours of adding the drug, it shuttles to the nucleus and can turn on gene expression. And so what we've gone through is to redo the initial studies to prove that this system similarly promotes definitive endoderm specification. And Nicole's shown beautifully that within 4 days of SOX17 activation, we get to the same sort of definitive endoderm population out the other side. So we're at this stage right here. And what we devised then was a series of more high throughput experiments to see which transcription factors could be expressed that would promote subsequent cell differentiation. So we had a list of candidates that we thought were a sure bet. like PDX1, and some that we thought might be a little bit more risky, like NGN3, and we just overexpressed them in human ES cells and used high-throughput real-time gene expression to try to understand what we had made at the end of the day. It turns out that our bets were off, and the ones that with the transcription factors that we thought were a sure bet to promote cell differentiation did very little, and ones where our colleagues thought would never work actually promoted differentiation, which we think is quite exciting. So I'll show you a little bit of that data now just to try to convince you that we're making some headway. So what you're looking at here are some of these populations of cells where we've overexpressed NGN3 in either the background of an undifferentiated human ES cell or in a human ES cell that's expressed SOX17. And hopefully what you can appreciate is this big change in morphology that we see. So over time, by somewhere between 8 and 12 days, we see a pretty overt change in cell morphology when we overexpress NGN3 in these cells. This is just a high magnification view of these cells where you can see the accumulation of some large cytoplasmic vacuoles and also can appreciate the large cytoplasmic to nuclear ratio of these cells. And we just confirmed by antibody staining that our transgene was actually positive in these cells and was being overexpressed and that functional protein was being made. And then Nicole went through and looked at a variety of different markers and did a couple of very interesting things. And so what we did was we compared human ES cells that were overexpressing NGN3 to our definitive endoderm cells that were overexpressing NGN3 to the gold standard growth factor protocols that were out there in the field. And so this is a bunch of work that was pioneered by Kevin Demood and his colleagues using a variety of cocktails of growth factors, and this is really what the field right now sees as the gold standard of differentiation. And so we wanted to say, what do our cells look like over time, and how do they compare to these other protocols? And so here the red lines are the double transgenic cell types, and hopefully what you can appreciate is that the markers of definitive endoderm go down over time, and that we actually see induction of a variety of pancreas markers that come on and then come off again. So we see peaks of NKX 2.2, peaks of NKX 6.1, we see PDX1 induction over the other ones, and some PAX4 coming on. However, the end of the day, after 16 days of differentiation, what we were really looking for is something that might look like a beta cell or like a pancreatic cell type. And so, of course, we assayed for levels of hormone expression and also markers of exocrine pancreas, because our goal in doing this was to try to be specific. And what we were thinking was that we might be able to promote differentiation in a bit more of a specific manner compared to growth factor cocktails or anything of the matter. So what we can see is that transgene expression was in fact able to promote more insulin gene expression than the green line, which is the growth factor mediated protocols in direct comparison, and that there was far less glucagon and even somatostatin expression in our cultures versus those. And quite surprisingly, we hadn't realized how many markers of exocrine pancreas were being induced in the growth factor protocols, and this wasn't actually happening using transcription factor mediated differentiation. So in summary, an overview and a little bit of data from that starting in my lab, where we're trying to understand how cell fate is governed. And so we really think that this reciprocity between intra and extracellular signals, how cells interact with each other, how they receive and send signals is quite understanding. And what we're hoping is that by using models to better understand cell fate, that we'll be able to get closer to generating functional cell types for therapeutic application.
Okay, so entertain some questions. Mr. Vandergoy.
What was the control, or were there controls for the Zen and mouse ES cell co-cultures? But do trophoblast cells not induce cardiac? If you just increase the number of mouse ES cells, you hasten cardiac differentiation?
So we haven't done the trophoblast co-culture experiments, but we think those are actually quite intriguing. And we actually want to do some trophoblast Zen co-cultures, but that's a different story. The controls that we did in all of those experiments were, of course, plus minus LIF, and also the presence of MEFs instead of Zen cells. And so, actually in the case of the human cells, we did co-culture with MEFs with undifferentiated cells and actually with SOX17 cells too. So we tried to control for both cell contact, cell density, and also the background media that everything was in. So they were always in the same basal media. The only difference was the cell type that was conditioning it. So the student, that was working on the experiment did do cell counts. And so what we do know is that there wasn't an actual big change in cell proliferation or the amount of cells. So we know that following 4 to 6 days of co-culture, the cells stop cycling actively and start differentiating instead. And so what we didn't appreciate was a big change in cell cycling. What we think instead is the intracellular signaling in response to the signals being emitted by the Zen cells.
Nick?
To what extent do you think some of these observations are selective in nature as opposed to effective?
For which? So for the...
Any of the lineages you're seeing, as you know from experience, it's very Darwinian culture. So when you add things in and you get percentage of things, you're really not going to survive an onslaught of the factors you've added in. So my question is, how many things do you think are you're propagating pre-existing things?
Absolutely.
Proliferation and then it gets exhausted versus actually changing the state of the cell.
Absolutely, and that's a great question. So, and that is actually can be addressed, I think, with some of the data that I didn't show you. So one of the sure shots we thought was we figured we'd overexpress PDX1 in the definitive endoderm, and absolutely we'd make some pancreas. There's no way. Derek's shown beautifully, everything's derived from PDX1 cells. PDX1 is the key to everything. When we overexpress PDX1 in our cells, what we see is no induction of any other markers except insulin. So essentially what we're seeing is we're adding a transcription factor, it's binding to one of its biggest promoters, that's its downstream target, and it's inducing insulin expression. What we've tried to do was carefully look at turning on other switches that are actually indicative of development happening in the dish. And so by looking for transcription factors that should be on between becoming definitive endoderm and being an insulin-positive cell, can we see something temporally happening between A and B that might actually tell us that differentiation was happening? And so we have several examples where we just see transcription factors binding to big targets and not really promoting differentiation like we would expect to see. And then the example of NGN3 where we actually see peaks and valleys of different genes coming on and coming off again, where we're actually a little bit more excited to follow through on those and try to figure out if we're actually making a differentiated cell type out the other end. And we also have the benefit of working with Dave hESC, who has these beautiful animal models where we can take our cells, which we think are progenitors, put them in vivo, ask if they can make differentiated cell types, ask if they could be functional. So we have a variety of animal models, and that's where we're going with it, is to try to now take these cells in vivo and understand if what we think we've made is actually a functional cell type and how it will behave in the animal.
Well, if there's no other questions, please join me in thanking Dr. Seguin for her talk.